With this talk, I really want to show you, to explain why we started to do single-cell in placenta, why we need it, because I think it's not very good when you blindly follow the trend and start to single-cell sequence everything. You should clearly understand why it is required for your scientific problem. And since we have quite a broad audience, I also included some slides which are explaining what kind of problems single-cell data introduces. And I will try to show you our preliminary results, which hasn't published yet, but I hope they will be soon. So I will start with what is placenta. That's actually a temporary organ linking mother and child, and it has two components, maternal and fetal. And during 9 months, it undergoes really major molecular, structural, and functional rearrangements. I might say that human placenta is really unique. You cannot use animal models because the fetal sites invade maternal sites to the extent which is never found in any other animals. Some people say that it's evolutionary because a lot of oxygen is needed for the human brain development. And the placenta acts as lungs, kidney, and different other organs for the fetus. It produces hormones to help maintain the pregnancy, and it protects the fetus from the mother immune system. But unfortunately, there are a number of diseases associated with placenta development. One is preeclampsia, and it affects up to 10% of women worldwide, unfortunately, and it's characterized by high blood pressure and protein in urine, and high blood pressure can lead to even seizures and possibly severe bleeding. It starts to manifest during the first trimester. That's why actually you cannot tell whether a woman is going to develop preeclampsia in early stages. And unfortunately, the cause is still not clear, but we know that placenta plays the major role. So the treatment is actually kind of non-existent, because usually you do a cesarean section on the earlier stages to just stop symptoms, but if it's not possible, you just use some kind of supportive medication to lower blood pressure. I try to show the pregnancy timeline with this plot, and the preeclampsia symptoms start to manifest somewhere after the week 20. And week 34 is when we decide whether it was early onset or late onset disease. So when the symptoms shows before week 34, it's early one. And in the darker colors, I tried to show when actually we get the majority of samples. Because unfortunately, here on the second trimester, we almost never get the samples, because it's too late to do an abortion and it's too early to do a delivery, because even if there are some complications, doctors try to prolong the pregnancy as much as possible. And then we have early samples, late onset samples, and normal samples. And you can see that the intersection between normal and complicated and the rest is super small. So for this, we do not have controls, because if we get sample without preeclampsia on this during this period, it's no longer considered normal, because something happened which made doctors to, for example, do a cesarean section. And here we don't know whether the woman is going to develop preeclampsia. That's why we cannot also say that it's a true control or a disease. So with this plot, I wanted to illustrate that actually late onset samples are sometimes super similar to the controls, because at this point placenta is already a dying organ, and all the signals which can be related to preeclampsia or any other disease are hidden within the major processes of apoptosis, for example.
So this is the plot from our study, and I want to explain you how we get a lot of false negatives in all these papers you can find in the public. Because let's say you have samples here, and you compare and you do differential expression analysis, and you say, okay, we don't have difference, then probably this gene is not responsible for a disease. But in reality, since you don't have any samples in between, you don't know what is going on. So another thing is we have false positives due to the studies being performed on the different stages of development of the placenta. If, for example, you try to compare early onset in red and healthy ones, of course, for this particular gene, you will mark him as differentially expressed. But if we take only early onset, we kind of already see a trend that the expression is lower and lower with the later stages of development or gestational age. And here the expression on the first trimester, and probably somewhere in between we have this increase, and we don't know, but we want to model it. That's why we need to have better data sets, and at some point we thought, okay, maybe if we incorporate more data, we will get better results, we will be able to model what is happening in between, we will be able to remove all these false positives. And that is also complicated by very interesting placenta structure, because we have fetal side and maternal side, and fetal side invade maternal side with this villi, which eventually are bathing in the mother's blood and supplying the child with nutrients. So we have these problems with false negatives, positives, then placenta structure, and we tried to integrate a lot of different data sets, because it actually has a lot of advantages, although do not forget that we have disadvantages as well, because if you get one bad data set in your compendium, your efforts are ruined, or you can just over-normalize when using different technologies, even different sequencing technologies. And if I were to report our findings, like let's say we integrated 300 samples, it looks amazing, but in reality you can see that we cannot cover all the tissues, we cannot cover all the trimesters. For the first one and the second one we have a super small amount of samples, and we had to kick a lot of studies because they were simply of poor quality. Well, our PhD student in our lab doing this work now, and he managed to produce quite a nice plot, and he managed actually to separate all these tissues and trimester from all these dozens of studies. And if you try to plot the same on a T-SNP, the previous one was a PCA, you can even see that the same tissues are separated by trimester here and here. But we found that still some tissues that are supposed to be super different, they happened in the same cluster, and we were thinking how we can explain that, and we tried to deconvolute expression profiles to see the cells composition in each tissue, and the picture we saw was very surprising. It was all over the place. We didn't see the samples to be homogeneous, and the question was, what can be used to improve these results or to kick out bad samples? And what is bad? What is a normal cell composition in a given tissue? What is abnormal? If you want to compare our at least known controls with the new samples pre-eclamptic ones, we need to decide whether we need to kick these particular samples from this tissue. And it happened that building this reference data is super complicated, and we were not able to find anything we can use, at least when we started to do it, because if you take cancer studies, you will find a lot of single-cell sequencing, but in terms of placenta, I think up to now maybe 5 articles were published, but they are super small, like 2 or 3 samples per article, and not from all the stages of development. That's how we joined Pediatric Cell Atlas initiative, which is a sister project of Human Cell Atlas, and tries to define how the biology of human works on different landscapes, differentiation, perturbations like environment, or maybe some infectious agents, and of course the development. And you probably saw this picture. I just wanted to show it because there is an important concept. So this kind of protocol, which is called dropset, is based on the microfluidics, which operates with tiny amounts of liquids flowing in the very narrow channels. And when the cell meets a barcode, in yellow we have the oil, and in blue we have water, it creates a droplet. And this technique is super high throughput, but it has its own drawbacks. For example, we can have an empty droplet, or sometimes several cells happen in the one droplet, and this event creates double the amount of genetic material, so we need to remove it, and we need to account for these things. And I really love this picture. It's totally applicable to placenta because it has super complicated structure, but it's about cancer. And if previously we could obtain only this very, very blurry picture, now we can actually separate and try to see whether, for example, for this particular situation, we need to apply several medications to target several different subpopulations of cancer cells, which is very synergetic with placenta, because a lot of people say we have a lot of in common with cancer because it's highly proliferative, it has a really short time period when it needs to develop and function. Another thing which we have to fight also in our studies is the complex structure of single cell data. If you remember how distribution of expression looks for sequencing or for microarrays, it's much more normal, it's more like bell-shaped, and here we have a very complex distribution. Probably these genes are maybe some housekeeping, these are actually doing something. Then we have zero inflation. It's an event when you have a cell with a lot of genes expressed as 0, and finally we have some noise which we ideally want to remove. So that's why you can see a lot of papers, a lot of technical papers that trying to fight this and trying to actually explain what is happening. I mean, biologically, because zero inflation can be because of a dropout event during reverse transcription, or maybe because of transcriptional bursting. I didn't even know about bursting before I started doing single cell sequencing, because I thought that it's kind of a uniform expression of genes in a cell, but no, it happens in a short burst. And actually, when we get our observed expression, it doesn't mean we can use it right away, because we have a lot of compounded variables, because our cell of interest can be on a different differentiation stage, it can have different stage of the cell cycle, and finally it can be on different stage of apoptosis. And if we are interested in this, for example, we need to account for these confounded variables and some kind of transform our observed data, so we can only after that finally use it to, for example, de novo identify new cells. And if I were to say what kind of two different types of analysis exist now in many single cell studies, first is of course clustering of cell types, and the second one is different development trajectories, and in our study we tried to do all of them. Well, it was hard to put everything on one picture, but I just wanted to show what kind of pipeline we defined for ourselves. First, it consists of very well-known tools like Zira. Then we use some less known but very good for our problem. Then we use a tool for Bayesian network learning. We were working together with collaborators from Poland because we cannot rely on public markers if you want to define new single cell map of placenta, and if you want to define new subtypes. And there are very placenta-specific problems, but it might be also interesting for people who are working in cancer, because actually in placenta you have cells of different genetic origin, because you have a mother cell and a child cell. And if a child is a male, it's easy to differentiate because you can check the Y chromosome genes expression. But what if mother, like obviously female, and child is also female? How to separate these cells if you're not doing genetic genotyping of the child? So it happens that there are some solutions for this, and people are trying to call SNPs from RNA sequencing data. It's super complicated and I'm not yet sure whether it really works well on our data, but we are working in that direction. So that's a super typical T-SNP plot you see in all the studies, but actually somehow UMAP works better for us. And from this plot, the question is, you see this huge cluster of the cells, those are actually trophoblasts, and you see this subcluster. So the question, whether should, maybe that's different subtypes of a trophoblast, maybe we should do a very great publication, some novel findings, but it happens that if you, it was actually aggregation of 3 different samples, and first I thought that probably we did something wrong with this integration, with the aggregation of samples, but then I realized that actually we had 2 female fetuses and 1 male. So it's not because the integration is wrong, it's because the sex of the child influences placenta expression so much that probably we should not even do the integration, and we should separate those samples into 2 different categories. Eventually, somehow we got this picture after a lot of work, because bioinformatician is not enough to get this, because you need to understand the biology very precisely, because unfortunately there is no tool that will just assign labels to your clusters, because you need to check whether it's a real cluster, it's not some kind of artifact. And what next we tried to do, since the huge cluster of trophoblasts most probably represent several different cell subpopulations, we tried to look closer only on these cells, because trophoblasts, even on the 3rd trimester of pregnancy, are still not differentiating, and they differentiate into several subtypes of cells, one of which is extravillous cytotrophoblasts, which is the most invasive cell type, and some theories say that this can account for preeclampsia development because of the failure to invade into a maternal site. So if we are able to say that this particular trophoblast subpopulation, for example, in our case, we really believe it's a cluster 4, which is responsible for regulation of placenta cell invasion. If we can infer this cell subtype, and when we get the samples on preeclampsia or similar diseases, we are no longer lost in all these signals. We can actually try to see whether there are these cells in the complicated pregnancy, and if anything is wrong with them. Or maybe there are actually several different diseases, not one, because we have early onset, late onset, we have severe preeclampsia, we have a mild one, and you should not think that this is the same biological process. So I don't have a conclusion because we are still doing a lot of work on that, and you never have end when you are trying to create a reference, not answering very particular biological question, and we would like also do some ligand receptor interactions. This plot I also got from our data, and of course differentiation trajectories, especially on the subclusters we managed to get from this huge cluster of trophoblasts. And I hope maybe sometime later I can finally give you a reference map. So I also would like to acknowledge people who are working in this project, because we get samples from Austria, sequence in Germany, and then analyze in Ukraine in this kind of case, which I really like, because coming from a computer science field, I feel that it's impossible for me to get all the biological questions right. And the last slide I want to show is the bioinformatics school we are going to do in autumn in Poland. This will be the fourth year in a row. We applied for additional funding, and if everything is successful, we will be able to provide lodging, food, everything for a very, very small registration fee. It's still open till 26th of July, and I really encourage you to apply, and you can talk to some people who already visited us and ask their impressions. And I really would like to thank again Hans and Arsene to organize this super